what I'd like to talk to you is a topic that's now very important in most of our projects, both on rat, and we've recently started also on human doing this this stuff. That's cell-specific cholinergic modulation of neocortical neurons, and I've added now and synaptic microcircuits, because we're not only looking at single cells and their excitability, but also changes in synaptic transmission at these neurons.
Thanks everyone for joining the next installment of the Blue Brain seminar series. And it's really a great pleasure and an honor to welcome Dirk Feldmeyer as the next speaker. Dirk is a notable slice electrophysiologist. So he worked at the Max Planck Institute with Bert Sackmann, also cross paths potentially with Henry and Carl, who are here as part of the audience. And he did some seminal studies to unravel the synaptic connectivity, mainly between layer 4 spiny stellets and layer 2-3 pyramidal cells, and then looking at other parts of the microcircuitry in layer 4 and layer 6. And more recently, he's also started looking at the effects of neuromodulators on the cellular and synaptic and potentially network effects in the neocortex. Without much ado, let's listen to Dirk's latest findings.
Thank you very much for the introduction, Srikant. So there's really a large number of neuromodulators released in the neocortex, and as you can see in this table, can you see that table? It's just you need to... Yeah, okay. As you can see in that table, the major ones are acetylcholine, noradrenaline, dopamine, and so on. And I've just listed the receptor types and the sources in the brain where they actually come from and where they project to the neocortex. Acetylcholine is listed first, and it is, among other receptors, special because it actually falls into two groups. They're the nicotinic receptor types, which are just ligand-gated ion channels composed of 5 subunits, and the G protein coupled receptors. And I will be talking about both, but focusing on the effects of muscarinic receptors, which have received slightly less attention than those of nicotinic receptors. So these are the muscarinic receptor subtypes we have in overall, mainly, so that is not only in the neocortex, but everywhere. They are the muscarinic receptor types of the M1 group, that's M1, M2, M3, and M5, and you can basically see they are actually coupled to G proteins. They're, the most important thing is that they can actually deactivate calcium channels, the L-type calcium channels, and the N and PQ type calcium channels, which are especially important because they are located at the synapse. And this they do via the direct pathway, as you can see, because the beta gamma subunit actually diffuses directly to the N or PQ subunits, N and PQ channels, and reduces their release probability. Then we have the muscarinic receptor types of the M2 and M4 receptors, which are actually inhibitory G proteins. Later hyperpolarized neurons, and also inactivate calcium channels at the synapse. There are also so-called GS proteins, but those are not coupled to acetylcholine receptors. And we have at the bottom right, the nicotinic receptors, mostly permeable to calcium and sodium to different degrees, and they activate voltage-dependent calcium channels indirectly, and as well release calcium from internal stores. So we have here the nicotinic receptor subtypes, the alpha-7, which is a homomeric receptor, and the alpha-2 beta-4 receptors, which are generally 2 alpha-2 and 2 beta-4. The homomeric alpha-7 receptors show fast desensitization kinetics. They're highly permeable to sodium and calcium. And on the other hand, the heteromeric alpha-2 beta-4 receptors, it's alpha-4 beta-2 receptors, they are show very slow desensitization kinetics. They are permeable to sodium and calcium as well, but have a low calcium permeability. A slightly higher calcium permeability is present when there is an alpha-5 subunit present, as you can see below. Receptors have been found, acetylcholine receptors have been found at both pre- and post-synaptic sites in a synaptic microcircuit. You can see here at the presynaptic site, mostly in this review, it has been stated that there are M4 and M2 receptors. I will show you that this is not really the case in the neocortex, but you find the alpha-4 beta-2 receptors and the alpha-7 receptors at the synapse, and at the postsynaptic site, both M1, M3, M4, and M5 receptors, as well as the alpha-4 beta-2 receptors. Image is a highly general picture, and what you see here is certainly not the case at all neocortical synapse when what I'm going to show you in the next minutes. So a very important thing which we find important when studying neuromodulation on the whole is the fact that you have completely different concentration profiles for G protein coupled receptors and ligand-gated ion channels. So you see the first curve you see here is the dose response curve for a G protein coupled receptor. And as you can see, it's basically in the low micromolar range. The EC50 for these receptors is generally something around 10 micromolar or even less. If you look at ligand-gated ion channel, you can actually see that the dose response curve is in the high 100 to millimolar range. And you basically see when you have maximally activated the G protein coupled receptors, the ligand-gated ion channel just start to be activated. So they're really very far apart. So that means that G protein coupled receptors are already active at very low concentrations and may also be active at tonic concentrations. This is also something we found in our results with several neuromodulators. So this is here, the next slide. In the next slide, you can actually see some of the major channel types modulated by muscarinic receptors. And I've just highlighted here some of the potassium and calcium channels, which I'm talking about in slightly more detail Now, so this is for the potassium channel, the KSAP channels, the inward rectifier of the type 2 type and the type 3 type, and the the small conductance calcium channels and potassium channels, as well as the L-type channels and the synaptically located N and PQ type channels. So what are the sources, next slide, what are the sources of acetylcholine in the brain? So in the neocortex, that's mainly the nucleus basalis of Minard from the basal forebrain. You can see here the red projections in the figure that come from the basal forebrain and project into all cortical layers from top to bottom. That's completely different, for example, from dopamine, which projects generally to deep cortical layers. There's also another source of acetylcholine, which is intracortical. It's released from vasoactive intestinal peptide expressing neurons, at least a subgroup of them. But these don't need to concern us here because they innervate mostly interneurons in layers 2, 3, and 1. So next slide. So there are several different modes of release of acetylcholine. And the first one is a direct synaptic release that has been shown for layer 5 pyramidal cells, for example, that there are cholinergic synapses that produce fast depolarizations due to the postsynaptic permeability to sodium and calcium. And the next step is when you have a synapse, as you can see here, the calcium that actually escapes the synaptic cleft may spill over onto other synaptic contacts, in this example, a glutamatergic synapse containing a muscarinic receptor that's then activated by the low concentrations that escapes the synaptic cleft. There's also the possibility of volume release. As you can see in the next slide, where there is not really a synapse established, but a cholinergic bouton is somewhat away from the presynaptic terminal or the postsynaptic cell, but the concentration achieved in their vicinity is still high enough to activate a muscarinic receptor. And something I'm not going to talk about here, but for sake of completeness, for some neuromodulators, here in this case adenosine, which we studied in detail, there is also a release from both glia, pre and postsynaptic cell via metabolism of ATP and adenosine. So adenosine is actually generated intracortically by non-vesicular release. So going back to acetylcholine, so we have these different modes I've just described for different transmitters. I've just listed that here. Extracortical afferents may come for glutamate, certainly acetylcholine, serotonin. The volume transmission is certainly a case for dopamine, noradrenaline, but also acetylcholine and serotonin. We have spillover at all synapses. You can find in the neocortex glutamate, GABA, and acetylcholine, and as I mentioned, adenosine for non-vesicular release. So the general hypothesis for acetylcholine in the neocortex is that it's a powerful excitatory transmitter. And that has been shown in a number of publications where application of high concentrations of acetylcholine increase the firing rate or even induce firing, in this case in pyramidal cells. However, I'm going to show you that this is not the whole picture. So the first study is somewhat older, so we have published that about 10 years ago. And this is the example of how layer 4 spiny neurons in the neocortex, so in the somatosensory barrel cortex, are modulated by acetylcholine. And as you can see in panels, in the panel C, an application of acetylcholine here for a time of about 40 seconds causes marked hyperpolarization. This one is actually more than 5 millivolts, and this is a really long persistent hyperpolarization, because even if you do it over minutes, it's actually not decreasing, so there is no desensitization. The effect is purely muscarinic. As you can see in the top row in panel D, there is no nicotine response, and the response is also blocked completely by atropine, arguing that this is really mediated by a G protein coupled receptor. So we were looking at the effects of on the excitability of these cells. So what acetylcholine does here is it increases the real base current. So it basically is decrease the input resistance of these cells. And as you can see here with the puff application, it can basically block high frequency firing in these cells markedly. You just apply briefly acetylcholine, and you can see very nicely. That's the next slide, sorry. You can see in C very nicely how the action potential firing is reduced. So just briefly in A, you can see the reduction, the increase in the real base current, probably mediated by a decrease, by an increase in the calcium, in the potassium conductance, and this causes reduction in action potential firing. So the next slide, you can actually see at what concentrations that's mediated. Here we found that the EC50 for this effect is basically 6.5 micromolar. That's very low. So that's clearly well below any concentration that's achieved in synaptic contact. And the effect is really direct, because applying TTX and GABA-Z CGP does not really affect the hyperpolarization in these cells. So we have a G protein mediated hyperpolarization in these layer 4 spiny neurons, meaning that acetylcholine is in the position to decrease the excitability of cells, not as has been previously assumed that it's only there to increase excitability. So, but I want you to bear in mind, the effects are really done at low concentrations. So what you can see in the next slide is actually that the effect of acetylcholine is layer specific. So in panel A, which shows pyramidal cell in layer 2-3, you can find normally a depolarization, which is occasionally preceded by hyperpolarization. Next, yeah, you can see that for B and in panel B and H. You can see the small transient hyperpolarization, which then develops into a depolarization, but that's not the case for all the cells. For the layer 4 spiny neurons, as I said, a persistent, strong hyperpolarization. And again, for the layer 5 pyramidal cells, we found generally a depolarization that in some cells was actually preceded by a short hyperpolarization. It has been shown previously, next slide, that these layer 5 pyramidal cells show a strong hyperpolarization that's actually also exclusively mediated by muscarinic receptors. And as you can see here, that's the same, but on a different time scale we found. And that is actually mediated by calcium influx, because this finding is only found as next. So you can see here the experiment we did. So it was a large current step evoking action potentials. And then with the next application, can you click? You see again the hyperpolarization, which is here either marked by an arrow in the small in A or in B by the yellow circle. You see a hyperpolarization. As long as you keep depolarizing the cell before the acetylcholine application, you see a hyperpolarization. But if you don't do it, you see that in the subsequent traces, you basically lose the hyperpolarization. And only at the end, you again see a hyperpolarization if the application is preceded by action potential firing. So this is due to the fact that you have calcium activated channels. So you need calcium influx before you see the hyperpolarization, and the channel responsible for that is the next click, is the small conductance potassium channel that's opened by M1 receptor activation. Next click. And going back to what we have here, I would like also to show that next slide, that the effect in layer 4 is, although it's different in different, the effect of acetylcholine, although it's different in different layers, is preserved between layers of different cortices. So we did the same type of recording and application, not only in somatosensory, but also in auditory and visual cortex, and the effect is here maintained. So next slide. So we here show the pharmacology for the receptor subtypes. So we can clearly show that in layer 5, it's mediated by an M1 receptor because the effect is blocked by pyrenzepine. But in layer 4, the hyperpolarization is basically blocked by tropicamide, an M4 receptor subtype, as you can see down there. The other receptor subtypes, so the M1 or M2 receptor antagonist, did not really markedly reduce the response. So next slide. So we also looked what is responsible for the ACH-induced hyperpolarization, and for this we used path application, as you can see in the small inset at B, and voltage clamp recordings. If you apply acetylcholine, you actually get an output current as seen in A, and that output current has a reversal potential around 90 millivolts, as you can see in B, and you can see actually the net current in C, which can be blocked, as shown in D, by barium, a hallmark of inward rectifying channels. But it's unfortunately the pharmacology we try to use to dissect the inward rectifying channel did not work because it's probably mediated by inward rectifier of the 3.3 type, but there's no at least at the time, there was no blocker available. So it's a inward rectifier of the 3 type that's opened by, next click, that's opened by M4 receptors, and the cause, the effect, next click, is the persistent hyperpolarization. So the inward rectifiers are responsible for persistent hyperpolarization. Next. So what we then looked at is which effect acetylcholine has actually on action potential firing. And as you can see in the top row, we fired at before and after acetylcholine application, 5 action potentials, and before acetylcholine, you see a marked development of an after hyperpolarization. And the current that's on the right, and in the present, in red, you can see that acetylcholine caused a reduction in this after hyperpolarization. That's also shown in the bottom graph. So the after hyperpolarization is reduced by 70%. And that's likely to be due to the closure of KM or potassium channels responsible for the slow after hyperpolarization. Next click. And that does not only result in a reduced action after hyperpolarization, but also in a frequency adaptation. Next. So this is not just a mere finding. Normally, if you work at subthreshold or at near thresholds firing, you actually see that in the absence of acetylcholine, you just get 2 action potentials with the same current, and then basically the current fades due to the after hyperpolarization. However, if the after hyperpolarization is blocked in acetylcholine, you have a more reliable action potential induction. And we found that quite markedly that with in an experiment where we increased the real base current with every successive pulse to get an action potential, and in C you can basically see that in the absence of acetylcholine, we really had to markedly increase the current, the rear base current, by up to 50%, whereas in acetylcholine, you reduce this rear base current markedly so that it was almost identical at all stimulus numbers. If that is clear, that is fine. Otherwise, I invite people to ask questions here at this point. Okay. So we then went on to study the effect of acetylcholine on synaptic transmission, and you can see in A that it actually reduces the PSP amplitude. So we did a puff application on synaptically connected layer 4 neurons, and not only did it reduce the amplitude, it also changed the membrane potential, as we would expect, but did not affect latency or rise time. However, if you use, next slide, if you look at the paired pulse behavior, you can actually see that the paired pulse behavior is altered. So you have a markedly smaller depression, and also the CV of the response increases, meaning that you probably have a reduction in the release probability. In C, you can see the effect of acetylcholine on the paired pulse ratio, which remains close to 0.7, but is significantly lower under controlled conditions. So actually, next click. So acetylcholine is here acting via the M4 receptor to close N or PQ type calcium channels at the synapse. And next click, that results in a reduction in the synaptic release probability. Next. So this effect can also be observed at another synaptic connection, which you can see here. That's the connection between layer 4 spinal neurons and layer 2-3 pyramidal cells. You have also a reduction in amplitude and an increase in the paired pulse ratio. So the amplitude is reduced by a factor of 30%. The CV increased from 0.3 to 0.5, and the paired pulse ratio from 0.7 to 0.9. So in summary, next slide. In summary, in contrast to what has been observed for pyramidal cells in other layers, in layer 4, acetylcholine induces a hyperpolarization mediated by M4 type receptors. The hyperpolarization is mediated by inward rectifier potassium channels. These channels also reduce the after hyperpolarization and thereby improve the temporal fidelity of action potential generation. And acetylcholine lowers the release probability as a synapse. Taken together, we propose that acetylcholine improves the signal-to-noise ratio at layer 4 spinal neuron synapses and increases their temporal fidelity. So we then went on to look at another layer which has not been studied in detail with respect to cholinergic modulation, that is cortical layer 6. And here we found two cell types when recording from different pyramidal cells. Either we found a hyperpolarization in a pyramidal cell, or we found a depolarization in the pyramidal cell. The firing properties were slightly different in these cells, and the EC50 for the hyperpolarization was slightly lower than that for the depolarization. So we went on to see whether there is a morphological difference between these cells. And but first we wanted to identify the receptor subtypes mediating this. Could you go to the next slide? So the depolarizing response we found was, the hyperpolarizing response here shown in voltage clamp as an outward current was actually blocked by tropicamide via M4 receptors, as you've seen for layer 4 spinal neurons. But the depolarizing response was blocked by pirenzepine completely, as you see in the bottom trace. So we had two cell types, one having M4 receptors and one that had M1 muscarinic acetylcholine receptors. These cells, next slide, differed markedly in their morphology. So the hyperpolarizing cells had a very dense axon. They projected actually over several barrel rows, as you can see here, and also in the show plot. And they were very dense with respect to their apical dendrites. Main projection areas were layer 3 and certainly layer 6. And the axonal length was markedly longer than that of the second group. And you have to bear in mind that you, in slice preparation, cut a lot of axons, so the factor of difference is certainly higher than that what we show here. So this cell type, the first one, is called the cortical layer 6 pyramidal cell, and they show always a hyperpolarizing response. Another cell type you can see below in the bottom row is the so-called corticothalamic cell. They project with their axons into the white matter, and they have a very narrow, very short axon that's generally projecting only up to layer 4. They show always a pronounced depolarization that is mediated by M1 receptors. And the two cell types differ, as I summarize here briefly, not only in their axonal properties, they also show different expression patterns. The most marked is here the expression pattern for the so-called language gene FOXP2, which is clearly present in the cortical cells, but not present in the corticothalamic cells, but not present in the cortical cells. Next slide. So, we, because we found that the EC50 for the depolarization was higher than that for the hyperpolarization, we tested again whether there would be a difference in the response if we use higher concentration of acetylcholine. This was also in light of earlier publications which showed a rather ubiquitous presence of nicotinic receptors in pyramidal cells. And when we applied acetylcholine at 30 micromolar in the both cell types, we basically could block the response almost entirely, although there was a very small response left, as you can see in A, in the presence of atropine. However, when you use, as in B, 1 millimolar acetylcholine, you can actually see a sizable response in the corticothalamic cells, but virtually no response in the cortical cells. And in C, you can see that the EC50 is actually, for this nicotinic response, is actually in the range I have shown previously. It's 1 millimolar, so 100-fold higher than that of muscarinic receptor subtypes. And in D, we tried to identify the receptor subtypes, and because we use the specific blocker DH beta E for alpha 2 beta 4 receptors, we could show that this not the alpha 7 receptor is the receptor mediating the depolarization in the corticothalamic cells. So just as a reminder and as an excursus, here are some examples from prefrontal cortex where they also did just puff application of acetylcholine onto different neuronal cell types. They actually found, apart from top layer 2-3, they always find cholinergic responses, which however differ, next click, which however differ in time course. And this is because the cells in layer 2-3 and layer 5, in this case in the prefrontal cortex, are have a cholinergic response mediated by alpha 7 receptors, and only in deep layers we found a response mediated by the alpha 4 beta 2 receptors. Next slide. So then we looked at effects on synaptic transmission, and both again on the cortical cells and the corticothalamic cells. And what we found was that in cortical cells, the synaptic transmission was significantly reduced, as you can see in E, and in the middle trace, so the gray trace in B. And when we apply tropicamide, the M4 muscarinic antagonist, the response came back to the control level, also visible in E. So the effect was a significant effect between control and acetylcholine, and it was actually a presynaptic effect, as you can see by the shift in the inter-event interval of the spontaneous events. So the situation was different for corticothalamic cells. We actually saw an increase, so that's in D, we saw an increase in the response, so in the spontaneous activity that again returned to normal when you block the response with a nicotinic antagonist. Shown, the effect is you can see that by comparing the black with the cyan trace, and also by looking at the curve in F. The effect is again a presynaptic effect. To look at that in more detail, we performed paired recordings between corticocortical and corticothalamic cells as presynaptic cells. And just as an introduction, I would just like to show that these cells differ in their synaptic properties. Connections with presynaptic cortical cells actually show paired pulse depression. As you can see on the left, connections with corticothalamic cells show a pronounced paired pulse facilitation, indicating that both cell types have markedly different synaptic properties. And here, just as an example, we have, next slide, please. We show the effects of acetylcholine on synaptic connections with cortical cells and corticothalamic cells. While we have tested quite a number of connections, I just show the examples of homotypic pairs between either cortical cells or corticothalamic cells. And in the third trace, where you mark EPSP amplitude, you can actually see that over time during acetylcholine application, shown in the gray bar, you can see a drop in the amplitude of the response, while for corticothalamic cells, you see an increase in the amplitude. So that can also be seen by the mean EPSP in control and ACH. So you see a marked reduction from the dark green to the light green curve in ACH for the cortical cells, but an increase in the response for corticothalamic cells. And that goes along with a change in paired pulse ratio. So the paired pulse ratio increases for cortical layer 6 pyramidal cells, but conversely so for the corticothalamic cells, and the failure rate changes also accordingly. We have an increase in the failure rate for cortical cells, but a decrease in for corticothalamic cells. This was, in our view, at first surprising, because if you have G protein coupled receptors of the Q and GIO type, which are basically the ones coupled to M1 and M4 receptors, you would expect to see a decrease in release probability at all synapses. But that was for our connections not the case, because next slide. So in the first case, the effect for cortical cells was indeed mediated by M4 type of receptors. So we did pharmacology here, as you can see in A. Control is black, acetylcholine is in gray, and acetylcholine plus the M4 antagonist tropicamide is actually shown in violet. So what you see is a reduction in amplitude and an increase in paired pulse ratio in acetylcholine, which is reversed if you apply the antagonist. And the effect is almost completely reversible if you scale the amplitude, as you can see there. Although there's still a minor residual component, probably due to incomplete washout. And you can see that amplitude, paired pulse ratio, and the coefficient of variation, as well as the failure rate, all return to their initial value once you apply tropicamide. So the effect of tropicamide on release probability is indicating that you have M4 receptors. Next slide. What is interesting is that there was a slight difference between the effects using in the absence of acetylcholine and in the presence of acetylcholine plus the antagonist. Next click. You can see actually this in an enlarged fashion. The violet trace is smaller than the trace in acetylcholine. This points to the fact that there is tonically present acetylcholine, which actually causes the effect on the so the slight depression of the amplitude when you block all receptors. We found that also for other neuromodulators, mostly, for example, for adenosine. Next, yeah. So here's the synaptic connections between two cortical thalamic neurons, and the situations you should see in B are first application of acetylcholine, that's in gray, then the application of pyranzepine, that's in this reddish color, and then the application of acetylcholine, pyranzepine, one receptor blocker, and mecamylamine as a general nicotinic receptor blocker. Because the response, as you can see, is generally very small, the traces look a little bit ragged. So this is just 200 microvolts as amplitude. And you see that the amplitude is enhanced in acetylcholine and doesn't really change much, especially not the paired pulse ratio, when you apply pyranzepine. That's in C, the red trace. But if you apply mecamylamine, you can see, especially in the paired pulse ratio, that it increases, again, indicating that the main effect here is really an effect via nicotinic receptors. We have now performed these experiments also in DH-beta-E, and it's just similar. So the most effect is the strong reduction in the paired pulse ratio, which returns to control in a nicotinic antagonist. Next slide. This is a summary of the effect. It actually shows the cortical cells contain muscarinic receptors of the M4 type, both at the soma and the synapse. So this is the orange cell, and they cause a reduction, so a hyperpolarization of the neuron, and also a reduction in the release probability of the cells. In contrast to that, corticothalamic cells contain at the soma muscarinic receptors of the M1 type, which cause a depolarization of the cell, but also receptors, nicotinic receptors at the synaptic contact of the alpha 4 beta 2 type, and these depolarize the presynaptic bouton, thereby increasing the synaptic release probability. And we could show at all synaptic connections we tested, both from corticothalamic cells to corticocortical cells and different types of interneurons, that the release probability of corticothalamic cells is invariably increased in the presence of acetylcholine. And that happens also already at very low concentrations of acetylcholine. In our case, we just applied 30 micromolar. It's probably because of the restricted space of the presynaptic bouton, where even a small current influx causes pronounced depolarization. So what I would like to point out that the differential distribution of muscarinic and nicotinic receptors causes different effects on both excitability of the neurons and release probability, and that a subcellular distribution, different distribution of the receptors can actually cause an increase in the release probability independent of the presence of a G protein. Next, this is just to summarize this briefly in text form. We see a hyperpolarizing cells in corticocortical and a depolarizing cell in corticothalamic cell. The effect is the effects on this excitability are mediated by M4 and M1 receptors, as well as alpha 4 beta 2 receptors in the cortical and corticothalamic cells, respectively. And the effect of acetylcholine on the release probability is decreasing in corticocortex cells, and that's mediated by M4 receptors. And in corticothalamic cells, the release probability is increased, but that's exclusively mediated by alpha 4 beta 2 receptors of nicotinic receptor subtypes. So on the whole, acetylcholine seems to improve the signal to noise ratio. Oh, sorry, that's disregard that. That's from sorry. So next slide. We also made a summary of all the effects, not only reported by us, but also by others. And when we look at muscarinic effects at all synaptic contacts that have been investigated so far, we also always see that muscarinic receptors at presynaptic boutons cause a reduction in the release probability. So when you observe in a few cases an increase in release probability in neocortical synapses, that can only be mediated by nicotinic receptor subtypes. Next. So I try to give a brief outlook on what we are doing now. So that's next slide. That's modulation of interneurons, and the challenge here is unfortunately that you have such a high variety of interneurons in the neocortex that broadly fall into the fast spiking and non-fast spiking group. And I've just shown here a few examples from the neocortex of both our data and that of other people's. And what we can show is that, next slide, that different types of fast spiking cells, here just in layer 4, this is a translaminar fast spiking interneuron and a large and a small basket cells. They show very different modulations, but especially in the case of acetylcholine, there is no marked cholinergic response in these neurons present. So fast spiking cells, on the whole, do not show a large cholinergic response, especially no nicotinic response, while they are very sensitive to dopamine and noradrenaline. The small response you see for serotonin is actually an indirect response when you block, when you apply serotonin in the presence of gabazine and TTX, the response disappears also completely. Next slide. We also looked at here just an example as a fast spiking cell. This is a neurogliaform cell and applied acetylcholine here, also at the low concentration. And what you see here for the different neuromodulators we used here, dopamine, noradrenaline, acetylcholine, and serotonin, there was only in the case, next click, only in the case of acetylcholine and serotonin, there were marked effects on the firing pattern, and the most marked ones were actually for acetylcholine, which increased the firing rate just enormously in control. Take into account that 30 micromolar is not very far away from the EC50, and is certainly much lower than anything you would, with anything you would activate a lot of nicotinic receptors. So acetylcholine is a profound modulator of most non-fast spiking cells, but has not much effect on fast spiking neurons. I wanted to show you in the next slide also some effects on our very recent data on human neurons, where we also found a profound effect similar to this one in non-fast spiking human interneurons. But unfortunately, I couldn't load the data because it was just too small. But so suffice to say, acetylcholine is also a similarly powerful neuromodulator in human cells, in human interneurons, in this case similar to the neurogliaform cell we found here, than it is in rat or mouse neurons. So with this, I would like to thank you and apologize very much for the problems that occurred in loading my presentation. And again, thank you, Srikant, for diligently helping me with the presentation and show all the figures in time. So I don't want to stop without mentioning the people involved. Next. These are for the layer 6 projects, Danjin Yang and Wang Xi, and Robert Günther and Gabriele Ratnikow, and for the layer 4 project, it's Emanuel Eggermann. We're continuing, especially now, the interneuron project and the human neuron projects, and we are in the framework of the HBP, next slide, well underway to study these cells. So I would also like to acknowledge the funding from the Research Center and the University Hospital Aachen, and the DFG in the research from Bakophon, and the Helmholtz Society, the Chinese Scholarship Council, and the Human Brain Project. Thank you very much for your attention.